Good morning, Year 13. So, what we're going to do today is we're going to finish off this little bit of the nervous system topic with talking about Pacinian corpuscles. Now, broadly, we've covered already the main body of the nerve, how that's activated. We've talked about the synapse and how that's activated. We talked about summation, which relates to the synapse. We've never really talked about how the nerves will be activated in the first place, how the neurons will be activated in the first place. We've never talked about receptors. Now, there are two types of receptors which you need to know a lot about, and those are the receptors of the skin, which are called Pacinian corpuscles, and we'll come to them in a second. And then there's some receptors in the retina of the eye, the rods and the cones, which we'll come to next lesson to completely finish everything off. So, before we do that, I'd like you to have a look at this starter, and I'd like you to pause me and work out which words, depolarization, repolarization, or hyperpolarization, goes with each sentence. So pause me now. So, increasing membrane permeability to sodium, I should say sodium ions to be fair, sodium ions causes depolarization. Opening the potassium voltage gated channels leads to repolarization. Opening sodium voltage gated channels leads to depolarization, that's the same point as the first one. Acetylcholine interacting with its receptor on the postsynaptic membrane leads to depolarization. And rising above the threshold potential leads to depolarization. So they're all depolarization except the second one, which is repolarization. Fine. So, today we're going to, as I said, talk about Pacinian corpuscles, and there's some objectives there. Basically, we've got to describe how Pacinian corpuscles work. So let's get started. Now, before we get started on that, there's already two receptors we've talked about, and we talked about them when we talked about the nervous control of the heart rate. Now, with the nervous control of the heart rate, we said that in order for the body to control the heart rate, we have to be able to determine blood pressure. Now, you need to know very little about these two types of receptors, but we need to mention them as part of the heart rate nervous control. Now, they're found in the aorta and carotid arteries, and they have two types of receptors. They have stretch receptors in the aorta and the carotid arteries, which detect blood pressure. Now again, we don't need to know much about this, but broadly, they're going to be detecting if the blood pressure is too high or too low because of how stretched or unstretched the arteries are. And they also have chemoreceptors to ensure the pH is the right level. And that's basically determining whether there is the right amount of carbon dioxide in the blood. The more carbon dioxide there is, the more acidic the blood will become. So fine. Let's move on. Now, this is the first one we need to know a lot about. These are touch receptors in your skin. And the first thing we need to know is the proper name for the touch receptors, the pressure receptors, perhaps a better way of putting it, in the skin. And they are, as I've said earlier, called Pacinian corpuscles. Pacinian, a very good Scrabble word. Now, that is a nice, straightforward name for something that is relatively complicated. So let's, if you pause me and get down these notes, and then we'll move on and we're going to need to learn about the structure of the Pacinian corpuscle. So, let's skip through this. They look like this, and you need to look at these pink things down the bottom here. Now this is a layer of the skin called the dermis. It's basically just under the top layer of your skin. So the top layer is called the epidermis, and that is basically full of dead cells. The dermis underneath is full of live cells, and some of these live cells make up Pacinian corpuscles. Now, they're surrounded by fat tissue. You don't know anything about that, to be honest, but this is what they will look like in situ. So in order to demonstrate the structure of the Pacinian corpuscle, we're going to have a look at a diagram. And the diagram looks like this. Now the diagram on the left is the one you want to focus on, and on the right is another electron micrograph. Now Pacinian corpuscles basically are a bundle of cells wrapped around a nerve fibre. So what we have is a single unmyelinated nerve fibre in here. And what we have around that is a load of connective tissue called a capsule. Now, this capsule is made of rings of connective tissue, as you can see. So you need to know this diagram, you need to be able to recognise that that is a Pacinian corpuscle. Now, what's going to be happening, as I'll show you in just a second, is that when we compress those rings of connective tissue, they're going to put pressure on the neuron in the middle, and I'll tell you how that's going to cause what we call a generator potential. So get that diagram down, please. That's an important one. You can simplify it a lot. It just needs to be some rings around a nerve fibre. So let's have a look in a bit more detail then. And I suppose you could have those labels. It's basically what we just described. Fine. Now, there are stretch-mediated sodium ion channels in the non-myelinated bit of the nerve, of the neuron. And what happens with stretch-mediated sodium ion channels is that when you put any kind of mechanical pressure on them, they basically bend, and as they bend, they open, and sodium ions are able to get through them. It's the same thing with pain receptors. Whenever somebody, I don't know, puts their hand on a pin or something, sits on a pin, we put pressure on neurons, and this causes these stretch receptors to open and activates the neuron. In this case, though, we are responding to the capsule, the rings of connective tissue being squashed. And as they're squashed, we are physically opening the sodium ion channel. Now, the sodium ions then rush in, and just like with the action potential, we're going to be going above a threshold, then we're going to go through it, basically an action potential. But this is called a generator potential because it's not caused by previous sodium ions having been entered, it's caused directly by some stimulus, in this case pressure, on the stretch-mediated sodium ion channels. Now, unlike an action potential, the stimulus intensity affects how much of a depolarization there is. So we don't just get to plus 40 and stop this time. It's not an all or nothing. The harder you press it, the more sodium ions enter, the higher up the generator potential goes. And the higher the generator potential, the more action potentials will be created in a certain amount of time. And remember, if you have lots and lots and lots of action potentials, that causes your brain to recognize something as a strong stimulus rather than a weak stimulus. Now, this is basically what I've just said. Now, broadly, we're going to use generator potentials for pretty much every receptor. So with Pacinian corpuscle, it's all about crushing that stretch-mediated sodium ion channel, allowing sodium ions in, and the more you press it, the more sodium ions get in. When we're talking about the stretch receptors in the aorta and the carotid arteries, that is exactly the same thing. If the blood vessel stretches, then the ring around it basically gets stretched as well, and that opens up stretch-mediated sodium ion channels. Obviously, when we get to the eye, it will be something slightly different because we're not talking about pressure there. But in every case, you are creating a generator potential, which will vary in strength with the strength of the stimulus and also with how long the stimulus lasts. Fine. Now, this is a very brief overview of exactly what we've just talked about. If you need any help with that, have a glance at that, but I'm hoping there won't be too much difficulty there. Basically said what we've just said. Right. Now, This is basically just exactly the same graph that we had before, but again, generated potential will increase further the more pressure you put on it. Fine. Now, there is a lot of questioning around this, and it tends to have to do with basically putting microelectrodes on a Pacinian corpuscle and testing how it works. So, relate the recordings in microelectrodes X and Y to the different pressures applied to the corpuscle. Now, hopefully, immediately you can look at this and say, right, well, X is on the non-myelinated nerve fibre within the Pacinian corpuscle, within the capsule, and Y is attached to the myelinated bit of the nerve fibre, which is going to be part of the neuron. So X is going to be creating generator potentials, which then cause action potentials in Y. So let's have a look at this. Have a look at light pressure. What can you see here? In X, clearly the generator potential is not very strong. So we're looking at only getting up to minus 50 from minus 60. Now, that only pushes the Y up to minus 50 as well, and that clearly is not enough to create a threshold, because you can see there is no action potential here. Basically, we haven't reached the threshold, and so what will happen? No action potential will pass along this neuron, and so you won't feel anything. So every time a piece of dust lands on your skin, you have this happen with light pressure. But because it's so light, the generator potential isn't enough to cause the threshold to be reached, so you get no action potentials, so your brain never knows about it, nor does any other part of the nervous system. Medium pressure, by which I'm talking about something you can feel, but it's not very strong. Well, now you can see, again, we've gone slightly up, even a little bit further by the generator potential, just over minus 40. And so we've gone over the old threshold. And so you can see an action potential. In fact, 2. So what is the difference between medium pressure and heavy pressure? Well, with medium pressure we only get a small increase in generator potential. With heavy pressure we get a large increase in the generator potential. And what effect does that have on the action potentials? Does it increase the size of the action potentials? No, but it increases the number of action potentials. And these kind of experiments where you literally get electrodes and detect the electricity enable us to understand the idea of all or nothing, the idea that you can't get big or small action potentials, and also the idea that if you have a strong stimulus, you get lots of action potentials. And this is what you can see here, and you'll see a lot of questions about this, one of which I'm about to show you. If you didn't understand that, there's some notes there. Fine. Now, this is a classic exam question, and it's pretty much what we've just talked about. A biologist investigated stimulation of a Pacinian corpuscle in the fingertip. She used her microelectrodes, we talked about them, to measure the maximum membrane potential. The figure below shows Pacinian corpuscle, its central neuron, and the position of the microelectrode. So it's exactly the same as what we just looked at. Instead of X and Y, it's P and Q. No difference. And there's your results. Now, I'm going to keep flicking backwards and forwards between these results. It might be worth taking a picture or screenshotting this and just putting it to one side so you can keep these results. That's probably the best way to do it. If you're on your phone, then it'll be a bit more difficult. You might have to scribble them down. If you're on a computer, just screenshot this now and put it to one side, and then have the questions on the other side. It's probably the best way to do it. And the questions are here. And I will let you carry on with this. So just pause me. So hopefully you've done that. It says 9 marks here, it's only 8 that I can see, but I might be wrong. So, A, explain how the resting potential of minus 70 millivolts is maintained. you will notice these questions just come up over and over and over and over again, and so we can immediately, hopefully, get that answer. How do we maintain a resting potential? We're looking at the membrane being more permeable to potassium ions and less permeable to sodium ions, or you can go into a bit more detail and you can talk about leak channels for the potassium ions and not for sodium ions. That's fine, and the sodium ions are actively transported out and potassium ions in. Now I would be going into a lot more detail than that, but those are the 2 marks you need, and I would anticipate everybody getting them. If you're still not understanding that, you have to understand this comes up pretty much every nervous system question, and it's pretty much this mark scheme, to be fair, particularly with a 2 marker. Sometimes you see a 3 or 4 marker, in which case they'll be more specific about sodium leak channels, 3 out, 2 in, this kind of an idea, but with a 2 marker you can get away with these 2 marks. Explain how applying pressure to the Pacinian corpuscle produces the change in membrane potential. So basically, why do we get a membrane potential change when we press the Pacinian corpuscle? Well, the idea that the pressure caused the membrane or the capsule to become deformed or stretched. The sodium ion channels in the membrane open and sodium ions move in. That's a mark. And greater pressure means more channels open and sodium ions enter. Now that's important because from our results we can see that the membrane potential increases as we press harder and harder. So the more pressure, the more channels open. You have to say that. It's important. C. The membrane potential at Q was the same whether medium or heavy pressure was applied to the fingertip. So basically, regardless of whether you do medium or heavy pressure, you get the same size action potential, and that's predictable. Threshold has been reached. Threshold causes maximal response, or the all or nothing principle. So the first thing is, why do we get an action potential at all? Why is it different to light pressure? Why is it not minus 70? Well, because we've reached the threshold. Second thing is, why is it not bigger for heavy pressure? Well, the all or nothing principle, the idea that there is no such thing as a big action potential and a small action potential. There is either an action potential or no action potential. The all or nothing principle. And then multiple sclerosis, in which parts of the myelin sheath surrounding neurons are destroyed. Explain how that results in slower responses. Again, a classic question in one form or another. Less or no saltatory conduction, less action potential or impulse unable to jump from node to node, and you need to do more depolarization over the length of the membrane. And that's the key point. You can't jump, and therefore you have to depolarize the whole membrane, which takes time. And that's the end of that. Now, Pacinian corpuscles as an idea is not difficult. You squash the thing, it opens the sodium ion channels, and that's it. These questions can be complicated, and I picked this one because it's classic. This mark scheme will be seen in loads and loads of exam question mark schemes. So if you don't understand that, it's worth going back, checking the points you didn't get, and adding them next time. Classic things people don't write. Greater pressure means more channels open. That's a classic one. The idea of the all or nothing response, and the idea of depolarization over length. Those are the 3 marks that people generally wouldn't get, so make sure you get them next time. And if you're not getting A and D pretty much 100% of the time now, you have to understand maintaining a resting potential and saltatory conduction, the old myelin sheath, comes up, am I going to say every year? No, every other year, probably. And who knows what comes up in your mock this year. So, hopefully that's not been too difficult. Any questions, as usual, contact me on Teams. I hope you have a very good day.